Welcome to my revision lesson on digestion.
This is relevant to the Edexcel iGCSE Biology Topic 2E Human Nutrition, but also covers Topic 4221 of AQA and Topic 7 of Cambridge iGCSE.
We will be looking at the structure of the digestive system from the mouth right down through to the anus. We're going to be looking at how the food moves by peristalsis. We're going to be looking at the digestive enzymes and what they break down and where, and the importance of bile.
Welcome to my revision lesson on digestion, from ingestion to egestion. Starting with the mouth then is ingestion, which is the taking in of food into the body. So here's Norman, he wants his nice dry biscuits and he starts to munch them up, masticate them using his teeth. And mastication is just the grinding of teeth, which is a physical digestion. But obviously he's trying to do this eating of the dried crackers and it's not going very far. So he starts to produce saliva. And saliva is a very important part of digestion in the mouth because not only does it lubricate the food, but it also has amylase in it. And amylase is very important for the breaking down of starch. So the amylase released in saliva starts to break down starch into maltose. So now Norman needs to swallow his food, and to do that he needs to use his tongue to push the food to the back of his mouth and down the top of the esophagus. And so then the esophagus takes over the pushing of the food down to the stomach. And the way that it does it is by using circular muscles which then contract and push the food like your hands pushing a rubber ball down a rubber tubing. And so the circular muscles contract, push behind the bolus of food, pushing that bolus of food down to the stomach. And this is peristalsis. The first port of call of the digestive system is the mouth. Food is taken into the body via the mouth, and this is ingestion. Your teeth then chew the food, otherwise known as mastication. This is a form of physical digestion as the food is being physically broken down. Saliva, released from the salivary gland, softens the food as it is chewed. It is slightly alkaline in pH, which helps the enzymes in the saliva. Amylase breaks starch down into maltose, and then maltase breaks down maltose into glucose. This is a chemical digestion. The tongue then pushes the food to the back of the mouth to be swallowed. The esophagus is a muscular tube which carries food from the mouth down to the stomach. This process is assisted by peristalsis, which is the contraction of the circular muscles of the esophagus behind the food, and this pushes the bolus of food down to the stomach.
So the food has now got down to the stomach, and in the stomach we have the churning of the food, and this is physical digestion. And we also have the addition of hydrochloric acid. Now hydrochloric acid is there to destroy any pathogens which might have entered with the food, and also it's very important for providing the optimum pH for the pepsin. And pepsin is a special enzyme which digests proteins down into amino acids. And the food is ready to be churned. And that makes a nice acidic chyme. Lovely. We've all known that when we've been sick. And this is going to stay in the stomach for several hours before it moves to the next stage. And it's held there by sphincters, which are special little circular muscles at the bottom of the stomach.
Hydrochloric acid is secreted in the stomach. This destroys pathogens that may have entered with the food. It also makes the environment acidic and so provides the optimum pH for pepsin, an enzyme secreted by the stomach. Pepsin is a protease enzyme that breaks protein down into amino acids. Please note here, as students often get confused, it is only the pepsin that does the digestion, not the hydrochloric acid. The acid is just there to provide an acidic pH for the enzyme pepsin to work in. The food is churned by the stomach, mixing the food with the stomach secretions and forming an acid chyme. The churning helps to physically digest the food, breaking it up into smaller pieces. Food stays in the stomach for several hours.
After it's been in the stomach for several hours, it then moves into the small intestine. Now, it's released into the small intestine by the opening of that sphincter at the bottom of the stomach, the pyloric sphincter. And what happens then is it moves into the small intestine, but it's acidic, which is not good. So we have to add something straight away to neutralise it. So in it goes into the small intestine. And to neutralise that acid chyme, we have to add a very special ingredient called bile. Bile's made in the liver and it's stored in the gallbladder. And as the food is released from the stomach into the first part of the small intestine, the bile is released down the bile duct from the gallbladder. And it has two very important roles. It neutralises that acid chyme and it also emulsifies fats, which means we're breaking down large fat globules into little tiny fat droplets. This increases the surface area for lipase to then work. Protease is also added, and that breaks down proteins into amino acids. Protease is the general term for any enzyme which breaks down proteins. Carbohydrases is the general term for any enzyme which breaks down carbohydrates. So it breaks down large, complex carbohydrates into the smaller monosaccharides or disaccharides. This breakdown of these food molecules is occurring in the duodenum. Digestion is the breakdown of large, insoluble food molecules into small, soluble ones, ready for them to be absorbed. And absorption is the taking up of those small, soluble food molecules from the small intestine into the blood. And that occurs in the ileum. Now, the ileum I have tried to represent by this highly folded piece of tissue paper, because the ileum has lots of finger-like projections called villi along it to increase the surface area, which means that diffusion can occur more rapidly and so absorption can occur more rapidly. The absorption of those food molecules is important because we then need to carry out assimilation, which is the build-up of larger biological molecules from small ones. So it could include using amino acids to build up new cell membranes to make new cells for growth.
When the food is released from the stomach, it enters the first part of the small intestine, called the duodenum, and this is where most of the chemical digestion occurs. Digestion is the breakdown of large insoluble food molecules into small soluble ones. Bile, which is made by the liver and stored in the gallbladder, is released down the bile duct into the duodenum as the acid chyme is released by the stomach. The bile has two important jobs. It is alkaline, so it neutralises the acid chyme. This is essential for digestion, as otherwise the acid conditions would denature digestive enzymes secreted by the pancreas. Bile also emulsifies lipids, which means it breaks down large lipid globules into small lipid droplets. This increases the surface area of the lipids so that lipase can then break them down more rapidly. After bile has been added, the digestive enzymes are added from the pancreas. The groups of enzymes added are lipases, proteases and carbohydrases, including amylase and maltase, which we met in the mouth. When I talk about groups of enzymes, they are grouped by what they break down, but each group contains lots of different enzymes. Like going to the butter aisle at a supermarket. It is filled with butters which all do the same job, but there are lots of different types. Spreading ones, wrapped blocks, those blended with buttermilk, lactose-free butter, salted, unsalted, and so on. Lipases break down lipids into fatty acids and glycerol. Proteases break down proteins into amino acids. And carbohydrases break down large carbohydrates into monosaccharides. Once the large, insoluble molecules have been broken down into small, soluble ones, they can be absorbed. Absorption is the movement of small, soluble food molecules from the small intestine into the blood. This happens by both diffusion and active transport. Most absorption occurs in the second part of the small intestine, called the ileum. This is the longest part of the small intestine, being 2 to 4 metres long, and it has special finger-like structures called villi along its internal surface to increase the rate of absorption. Villi, which is the plural, the singular is villus, are specially adapted for absorption in several ways. They provide a large surface area for rapid absorption, and the surface area is further increased by the microvilli on the outer surface of the villus wall cells, as shown in the diagram. The wall of a villus is just 1 cell thick, which ensures that there is only a short diffusion distance or a short distance across which active transport has to occur. There is a good network of blood capillaries which transport absorbed molecules away from the small intestine, therefore maintaining a steep diffusion gradient. There is an internal structure called a lacteal, which transports fatty acids and glycerol away from the small intestine in the lymph. Once the food molecules have been absorbed, they are used in assimilation. Assimilation is the movement of digested food molecules into the cells of the body where they are used. For example, glucose is used in respiration to provide energy, and amino acids are used to build new proteins.
Next, the food moves from the ileum into the large intestine. In the large intestine, it's very important that water is reabsorbed because we've added an awful lot of liquid to this food as it's gone through, and we're land-dwelling organisms, so we need to reabsorb as much water as possible. So the main job of the large intestine is to reabsorb the water back into the blood. But there's also a gut biome, which is very important, which are healthy bacteria, and they can help us digest some of the food and reabsorb vitamins from the food as well. So let's have a look. Here we go, some reabsorption of water going on in the large intestine. And so it goes, we do reabsorb as much as possible. If we have got a stomach problem, we could end up with diarrhea. And diarrhea is when it gets pushed through the large intestine so quickly that we can't reabsorb the water, and so it comes out very liquidy. And so once we've reabsorbed an awful lot of that water, it then moves into the rectum, which is the final part of the large intestine. And what happens is the rectum stores all of that undigested food. This food has never entered any of our cells. It hasn't gone through any metabolic reactions. So it is not an excretory waste. We must be careful about that. It has its own special word for getting rid of this from the body, and that's egestion. So egestion is the removal of undigested food from the body through the anus. So please do not get egestion and excretion mixed up. So here we have the rectum, and it's full of its undigested food, and it sits there until we're ready to go to the toilet. And most of us have got control over that, and so we will wait until we go to the toilet. And when we go to the toilet, we will release our sphincter, which is holding our anus shut. Remember, the sphincter is a circular muscle which will hold the anus shut. And when we're ready, what happens? Oh, I hope it doesn't work like that. But we go to the toilet and we ingest all of that undigested food down the toilet. And that is the end of the whole process of digestion, from ingestion to egestion.
The small intestine absorbs much of the water and most of the digested food. The remaining material in the intestines consists of water, bacteria, indigestible substances such as cellulose from plant cell walls, and cells which have been sloughed off. Fibre helps with this removal of old cells from the lining of the intestine. The material passes into the colon, which is the first part of the large intestine. It absorbs most of the remaining water and minerals. It also harbours our gut biome, which is very important to our health. It is now known that the gut biome not only helps with fermenting undigested food, but it interacts with the immune system. It helps with the producing of vitamins such as vitamin K. It triggers the release of hormones involved in fat storage and influences our mood, so we need to look after it, which means a healthy diet is important. After absorption of the water and minerals by the colon, a semi-solid waste material called faeces is formed. The faeces is stored in the rectum, the last part of the large intestine. When the rectum is full and the person is ready to go to the toilet, they release the faeces out of the body through the anus. This is egestion. Please do not get excretion and egestion mixed up. Excretion is the removal of metabolic waste from the body. The undigested food making up faeces has not entered your cells and so has not been involved in chemical reactions in your cells. Therefore, it is not a metabolic waste. It has just passed through your body through a series of tubes. Therefore, the removal of it is called egestion. Let's look at an example of an exam question on digestion. In digestion, large food molecules are broken down into small food molecules by enzymes. Use this information to complete the table, and this is awarded 5 marks. You need to cast your mind back to those pancreatic enzymes released into the duodenum and to what you learnt about amylase in saliva. If you remember, amylase breaks down starch molecules into maltose, which is then broken down into glucose by maltase. So this first answer for the first box is maltose. The name of the enzyme protease sounds very much like the molecule it breaks down, which is protein. So this goes into the next box. And the building blocks of protein are amino acids. Finally, lipids. These are emulsified by bile, but the enzyme that carries out the chemical digestion is lipase. And there are 2 products of lipids, fatty acids and glycerol, so you should put both of these in here. Part B then. The small food molecules can be absorbed into the blood by villi in the small intestine. Here there are 3 marks awarded. Give 3 ways in which villi are adapted to absorb small food molecules. Can you remember the diagram of the villus and how it is adapted for absorption of food from the ileum? First of all, it gives a large surface area to increase the rate of diffusion. And if you remember, the surface area is further increased by microvilli. The wall of the villus is only 1 cell thick, which gives it a short diffusion distance. There are also a lot of capillaries, so this maintains a steep diffusion gradient. If you remember that because they are carrying away those absorbed food substances. And then there's the lacteal down the centre to absorb digested lipids. Let's look at a second example of an exam question. The table contains names and descriptions of processes involved in the digestive system. Complete the table by filling in the missing names and descriptions. There are 5 marks for this question. Here you have to remember the definitions we went through in the lesson. Remember, it goes ingestion, digestion, absorption, assimilation and egestion. The first row is therefore ingestion. The definition of digestion is the breakdown of large insoluble food molecules into small soluble ones. Here it is important to mention both the size and the solubility. Then we have absorption, as this is when the small food molecules move from the small intestine into the blood. Careful with the spelling. Then assimilation is when small soluble food molecules move into the cells of the body where they are used. So the use of the small food molecules to build into larger molecules is part of assimilation. With the definition of egestion, you need to make sure you get the buzzwords in. Faeces, careful with the spelling again, and anus. So it is the removal of faeces from the body through the anus. Part B then. Describe the process of digestion in the mouth. 3 marks. There are 2 parts to this, mastication and the importance of the saliva in chemical digestion. Mastication breaks the food down into smaller pieces, so this is physical digestion. Saliva is involved in chemical digestion as it contains amylase and maltase. Amylase breaks down starch to maltose, and maltase breaks maltose down to glucose. Hopefully this has helped with your approach to answering the exam questions. Please do not forget the buzzwords. As you can see, they are very important to get awarded those marks. Make flashcards of them and keep them in topics, maybe using colour coding, but please keep going over them. This brings me to the end of my digestion revision lesson, and I hope you will come back for more lessons in the future.